Which elements make up carbohydrates and in what ratio? Carbo stands for carbon, and hydrate is water. So water is H2O. And so the ratio, the elements, first of all, are carbon, hydrogen, and oxygen, and the ratio is 1 carbon to 2 hydrogens to 1 oxygen. What elements make up lipids? Same thing, except you only have a little bit of O. And the ratio is just, lots of carbons and hydrogens with a tiny bit of oxygen. So for every carbon, you basically have 2 hydrogens, minus a little bit with some oxygens, but not very many oxygens. So if you saw C1000, H 1090, 1989 or something, and O 978, whatever, this would probably be a carbohydrate because that's about a 1-to-1 ratio with about double as many hydrogens. If you saw something like this, with only 12 oxygens, then that would be a lipid. Okay, the elements that make up proteins. Remember, in general, the elements in living things that I want you to know are TOMs. And so that's what proteins are minus the P. You take the P out and put it in the word protein. So the elements in proteins are CHONs, and the elements in nucleic acids are CHOMP, and I don't know why I circled the N. So it's the opposite with N versus P. What are the monomers of carbohydrates? Well, luckily for us, the monomers also start with mono, and so they are mono for 1, and then the carbohydrate word is saccharide. How do I move this down? Really, how do I move you down? Huh. All right, I'm going to pause you while I figure this out. Okay, so the monomers of triglycerides, and a triglyceride is a lipid, and the monomers are 2 things. The tri is for 3, and so that's 3 fatty acids. And it also has 1 glycerol. So 1 glycerol, 3 fatty acids. You don't want to confuse that with glycogen, which is a carb. And triglycerides look like this. There's a glycerol molecule, and then there's 3 fatty acid chains coming off of it. So that's that, and that is that. What are monomers of phospholipids? Same thing almost. You've got the glycerol and 2 fatty acids, but in this spot you've got a phosphate group. So glycerol, 1 glycerol, 2 fatty acids. So far almost the same thing as the triglycerides. And then there's this head, the part that's got a positive and a negative charge. So for lack of a better name right now, I'm going to call it the phosphate part. The monomers of proteins. So these are different from lipids because lipids don't really fit into a nice category. With proteins, all of the monomers are amino acids. And there are 20 different amino acids. The monomers of nucleic acids are nucleotides. The major functions of carbs. So there are 2 major functions. One is energy. And you have things like glucose that are quick energy, and then things like starch and glycogen, which are longer-term energy. And then there are carbohydrates that are structural, that actually make the body of a plant or help make the body of fungi. Distinguish between monosaccharides, disaccharides, and polysaccharides. First of all, how are they all the same? They're all the same because they have this word saccharide in them. And saccharide means sugar. So they're all sugars or carbohydrates, whether they're a simple sugar here or a complex sugar here. Monosaccharides are 1 sugar unit, disaccharides are 2 sugar units, and polysaccharides are many sugar units all bonded together. Distinguish between glycogen and starch, and how are they similar? They're similar because they are both polysaccharides. And one thing that you might be memorizing but forgetting the big picture is that polysaccharides are, oh, don't go away. Shoot. Okay, so I'll do that part again. So they're similar because they're polysaccharides. I think I clicked something on this pen by accident. Saccharides. And the thing that students forget sometimes is that polysaccharides are a type of carbohydrate. So if you have time to look at those organizers that I made for you, it would help probably organize your mind with this. Glycogen is the polysaccharide found in animals. Animals make it. And starch is the carbohydrate in plants. And another reason that they're similar is they both store energy. So when you have too much glucose, you change it into glycogen. And when a plant has too much glucose, they change it into starch. So we can use them later. Distinguish between cellulose and chitin, and how are they similar? Again, they are both polysaccharides. Which means they are both carbs, carbohydrates. And they're complex carbs, same with these. These are all complex carbs, they're all polysaccharides. And they don't store energy though, they are structural. So what does structural mean? Structural means they make the structure or the physical part of the body. So for cellulose, it makes the structure of a plant's cell wall. And for chitin, it makes a fungi's cell wall, or it makes the exoskeletons of crustaceans. I don't know if you spell it that way. And insects. I gotta assume arachnids are in there too. Okay, distinguish between cellulose and starch. How are they similar? Well, they're similar for some of the same reasons as above. They're both polysaccharides, which means that they are complex carbs, complex carbohydrates. And they're also similar because they're both found in plants. They're different because starch is used by plants for energy, and cellulose is used by plants for structure. Okay, from what structure are steroids made? They're all made from cholesterol. And so they are all these fused carbon rings. There's 4 carbon rings that are fused together. They look, let's see, something like this. I'm going to run out of space. And then they have whatever different side chains stuck on here. Why are steroids considered lipids? For one reason only, they are hydrophobic. The functions of phospholipids. So phospholipids are those adorable little cells. They look like this. They don't really look like that. They more look like this, glycogen, with a couple of fatty acids and this charged thing sticking up here. So anyway, the functions of phospholipids is that they make up the cell membrane. And that's a really important thing because we are studying life, and all living things are made out of cells, and all cells have a cell membrane. And lots of cells have internal membranes too. Functions of triglycerides, mainly what I want you to know here is that it's a long-term energy storage. So that's the function of fat or triglyceride. Another thing is that it can insulate the body. So if you think of a polar bear, they have a nice layer of blubber, and that insulates them. Could you call it blubber with a polar bear or just fat? Whales have blubber. Anyway, big layer of fat to keep them nice and warm. And then it cushions organs and stuff too. Some functions of steroids. So some steroids are hormones. So cortisol, testosterone, aldosterone, estrogen, those are hormones. So they regulate something in the body. Cholesterol is another one, that's actually also found in cell membranes with phospholipids, but there aren't quite as many of those. There are some vitamins that are steroids too. What enzymes are what type of macromolecule? They are almost always proteins. Occasionally RNA works as an enzyme, but 99.99% of them or whatever, I think, are proteins. Whoops. Hormones can be what type of macromolecules? Two things. Either they can be lipids, and when they are lipids, they are steroids, or they're protein-based. So an example of a hormone that's a protein would be insulin, and an example of a steroid or lipid hormone would be testosterone or estrogen. Describe the difference between the levels of structure of a protein. So there's the primary, the secondary, the tertiary, and the quaternary. The primary is just amino acid to amino acid to amino acid. It's the order or the sequence of amino acids in a polypeptide or a protein. So I'm going to put order of amino acids. The secondary structure is how these things make alpha helices or beta pleated sheets, and that's caused by hydrogen bonding. The third thing is how they fold over onto themselves. So three-dimensional folding. So 2 is also that, but with the tertiary structure, it's actually not close. So with the secondary structure, these interactions here are fairly close on the polypeptide chain. So it might be this amino acid with an amino acid down here. But with the three-dimensional folding, you've got those interactions, but then it comes over and it folds over on itself like this. So 3D folding, and there are hydrogen bonds, ionic bonds, covalent bonds involved in that 3D folding. And the quaternary structure is when you have 2 or more polypeptide chains coming together. So here's one, and then here's another, and when they interact together and bond together, that would be the protein, and that's the quaternary structure. Some proteins are done right here with the tertiary structure, and then some have the quaternary structure as well. Once they're done folding, you can call them a protein. A peptide bond is the bond between amino acids. So you have carbon, no, what is it? Nitrogen, carbon, carbon with an O. And so you've got a hydrogen here and an R group here. And that is bonded to nitrogen, carbon, carbon with an O. This bond right here separates out this amino acid from this amino acid, and that's a peptide bond. It's a type of covalent bond. And it's only named a peptide bond because of where it is, not because it's anything different physically than any of these other bonds. What's the functional groups in amino acids? This N group is the amino group, and the COO, and it should be bonded to an O usually. Let me show you that. So here's carbon, it's got a hydrogen on it. It's got the amino group, which should be NH2 or NH3. And then it's got this R group, and then it's got this carboxyl group, which is C double bond to O and then to OH. And so that's carboxyl. So it has 4 things: a hydrogen, an amino group, which is this, carboxyl group, which is this, and an R group. The function of DNA is to store genetic information. Uh-oh, my thing's not writing anymore. Okay, so the parts of a nucleotide, whoops. Ah. Okay, so there's a sugar, a phosphate, and a base. And I actually thought I could get this all done, but